So good afternoon. Sorry to get a little bit of a late start. The room was busy. So this is the education session on antibody-drug conjugates, future directions and opportunities. Welcome. I think this is a topic that really doesn't need an introduction. All you really needed to do was show up at the oral session this morning, which was, for those of you who weren't there, entirely devoted to antibody-drug conjugates and to HER2-low breast cancer, which is a phenotype which wouldn't even exist if it wasn't for antibody-drug conjugates. So, this is now a really impactful area. Since ADCs entered the clinical space, which was almost exactly 10 years ago with the approval of TDM1, I don't think any class of drug other than immunotherapy has had a bigger clinical impact than antibody-drug conjugates, which are now multiplied and are used widely across many cancer types. And I really think we've only just begun to scratch the surface of the potential for these drugs. It may be just me, but I think it's reasonably likely that in the very near future we're going to replace conventional chemotherapy altogether and just use sequential ADCs to deliver cytotoxic agents. But we clearly have a lot to learn. There are a number of questions that need to be answered. Are there other targets beyond the ones we have that can be exploited? Can we improve upon the payload and linker technology? Almost certainly we can. How do we mitigate some of the toxicities that we're seeing with the current generations of ADCs? And importantly, of course, how do we overcome resistance? So fortunately, we have three great speakers today to help us answer these questions. I will introduce each one as they come up. The first is Dr. Puja Sapra, who is the Senior Vice President of the Biologics Engineering and Targeted Delivery Group at AZ. She leads discovery and development of therapeutics there using novel delivery technologies. She has extensive experience in the ADC field and therefore is perfect to be telling us about bioengineering and mechanism of action of ADCs. Thank you, Puja. We'll have questions after each speaker.
Thank you, Dr. Kropp, for the kind introduction, and thanks to the organizers for the invitation here. So as this is an educational session, what I thought I'll do today is give you a history of antibody-drug conjugate development, and then go into some of the different components which are required to be put together in an ADC design, what we have learned as a field, and then take HER2 ADCs as an example of what has worked, compare and contrast the two HER2 ADCs, and take it from there what the future could look like. So simplistically, an antibody-drug conjugate is an antibody which is designed against a target antigen expressed on cancer cells or overexpressed on cancer cells. It is attached by a linker to a payload. The postulated mechanism of action of ADC is binding on the target cells. ADC binds to the cells. It's internalized by receptor-mediated endocytosis. Inside the cell, appropriate cleavage mechanisms break the linker and release the payload, which attacks the cells and causes cell death. Besides this mechanism, the warhead can also permeate outside the cell and can kill neighboring cells by so-called bystander activity, which we'll discuss more later. And also, ADCs may have inherent antibody-mediated mechanisms like ADCC, CDC, depending on the design. While this cartoon looks quite easy on the slide, what is important to realize that all these components of the design is extremely complicated, and researchers have to look at each aspect of the design of the molecule, starting from what target to choose from, what density to go for, what kind of internalization mechanisms, etc. And because of that, this complex modality has taken decades and decades of research to be at the stage where we are today. One of my first recollection of an ADC which entered into the clinic was a Lewis Y antigen conjugated to doxorubicin. And while that showed good promises in preclinical experiment, this drug did not work in the clinic. However, it really set the stage off that this concept is possible, and with improved linker designs and the chemistry, we can make next generation drugs. But it also led the researchers to think at that time that doxorubicin was a very low potency molecule, and we have to move into extremely high potency molecule to make ADCs effective. This leads to, obviously, to coming up with very high potency molecule like Mylotarg and Bespansa, which were a calicheamicin-based drug, were approved in hematological malignancies. But the inflection point in this field really came around 2011, 2013, where two drugs, Etcetras for Hodgkin's lymphoma and Kadcyla for breast cancer for solid tumor, were approved. And this was using microtubule inhibitor. This time really created enthusiasm in the field and draw a lot of attention from biotech, pharmaceuticals, and academia to invest in this modality. The fruits of which is what we have started to see in the last 3 years. We now have 12 ADCs approved, not just for breast cancer, but different solid tumors and hematological malignancies. It's important to realize that this is not just again one or two targets, which was always the fear. It's against multiple targets. It's also using multiple mechanisms of action of warheads. And what is also what is quite important is we are seeing broad and deep responses even in late lines. We are seeing complete responses, and most importantly, the emerging data on combination with checkpoint is what's going to be extremely exciting, and we are seeing evidence of two mechanisms combining very well. However, the field has suffered a lot of failures as well. We have more than 150 ADCs in the clinic right now. We have 55 molecules which have been discontinued, and a learning has emerged in the field. So what has years, decades and decades of research has really taught us? And in a big picture, it's really that this is a three-piece puzzle, and you have to think about all the three components of the ADC design when you're making the molecule to generate effective molecules. The few of the early generation ADC designs relied on just developing, for example, linker payload, and thinking that once we have a linker payload, we can put on any antibody and any target and it will work. And that's not the case. It really starts from what's your end game, which tumor type to go for, which target to go for, what mechanism, and what is the biology around it, and how to design those chemistry and linkers. So for choosing the target, very simplistically, what's emerging as a trend for the successful ADCs is target antigen density. Most of the ADCs which have been approved or are looking good in the clinic comes with high target density, like at least for solid tumors like HER2, folate receptor, TROP2. In heme malignancies, because of the accessibility of the tumors, lineage-restricted antigens and lineage-restricted antigens, less copy numbers may be fine, but protein density is very important. ADCs work because they don't bind. ADCs work because they internalize and kill the cell, and that's why internalization is a very important aspect in thinking of the design. While this may sound very simple, you can, in the same campaign of finding an antibody, you can find antibodies which have very different internalization kinetics, and that can make or break a good molecule. Today, we have technologies that right from the get-go, when we are screening from binders at the time of phage display or hybridomer technology, we can screen for what would be the best internalizing antibody and pick them from the get-go rather than doing those experiments later. The heterogeneity of tumor expression, it goes without saying that in solid tumors, we are going to see that. And this is a very important aspect to think about for ADCs. One way for ADC to work, overcome heterogeneity of expression, is by engineering in the linker technology a bystander component. And what that means is that when the payload comes off, it is cell permeable, has the ability to go from the target cell to a neighboring cell which may not express the target and kill it. All the successful ADCs today in the clinic in solid tumors, except for one, has this component built in. The experiment on the left is actually on the bottom of this. It shows you is example with Enhertu or TDXT, where the authors had put cells which mixed with HER2 expression, positive and HER2 negative, and showed that Enhertu could impact both components of the cells. And the last is normal tissue expression, which is also a very important component because you want to avoid on-target tox. However, again, it's not a one-size-fits-all story here. The same target may work with one kind of mechanism of action of payload, but may not work with a different kind of mechanism of action payload with a normal tissue expression. Example here is TROP2. We have a successful drug and approved drug in the clinic with TROP2, and another one which is in late stage looking promising. However, the same TROP2 ADC with the MMA conjugate was tried in the past, and it failed in the clinic because of significant on-target tox. In terms of payloads, the 3 payload mechanisms that have been successful so far in the clinic are DNA double-strand breakers, the number 1. These are extremely highly potent molecules. These comes from natural compounds, and the best examples here are the calicheamicin-based drug or PBD-based drug. They have been very successful in hematological malignancies, Mylotarg, Bespanza, Zenlanta, for example. However, because of the potency and because of the tox profile, they have had so far restricted therapeutic index in solid malignancies. The second class is the microtubule inhibitor. And this has been one of the most successful class so far in terms of the approvals. As you can see here, these molecules have been put in context of a cleavable linker or a non-cleavable linker. They have high potency as well. And most interestingly, there is emerging data with this class as well of synergy with checkpoint inhibitors. However, this class of molecules have also been limited by off-target tox, and in most cases, it has come from myelosuppression or peripheral neuropathy. And the third class, which is now seemed to be quite interesting and exciting, and a lot of data is coming from in this class and is being discussed in this symposium as well, is in the topoisomerase 1 inhibitor category. This class differs from the other two in the potency range. This falls in the mid to high potency, and what that has helped us with has been able to inject the antibody drug conjugate at a much higher dose in the clinic compared to what the first two classes was able to do. We can now go up to 5 to 6 to 10 milligram per kilogram in the clinic, which was not achievable in the first two classes. Also, with the topo platform, we have been able to load much higher drug-to-antibody ratio than the other two classes. And again, in this class also, we have started seeing the evidence of combination benefit with checkpoint inhibitors, and this is also which has been discussed in this conference as well. So, what about the conjugation chemistry and how we can conjugate these molecules even better? One of the, while we have made great success in the field of ADCs, there's still room for improvement. And ADCs are limited today by what is called, in most cases, by what is called off-target tox, which means that it's not that the on-target tox is what is limiting in the clinic, but the tox which is coming from the warhead is limiting it. And this comes because of two reasons. Either the payload is coming off in the circulation prematurely, or the ADC is non-specifically taken up by various cells in the body. So while we have improved our conjugation technologies a lot and tried to improve this therapeutic window, if the game changer in this field will be if we can even improve the conjugation chemistries to take it to the next level and widen the therapeutic index. Today, most of the ADCs are conjugated by what is called stochastic conjugation, which is relying on the native lysines and cysteines in the protein molecule to conjugate. There are about 80 or so lysines in an antibody molecule. And what you can end up with is a heterogeneous mixture. Each of these bars that you see could, in fact, be a drug in itself with its own pharmacokinetic and tox profile. Where the field is now going into is what is called site-specific conjugation technology, where you can engineer certain residues in the antibody molecule or use non-natural amino acid to precisely load the drugs into the antibody structure. This would lead to a homogeneous mixture. It will be a much better profile when it comes to manufacturing and analytics of the molecule, and could also lead to improved therapeutic index. So far, although I would say all the ADCs that are approved rely on stochastic conjugation, which is pretty good, but what you're gonna see in the clinic and what is emerging is a lot of molecules which use site-specific technology and would be the next wave to see how they fare in the clinic. On the bottom, I'm showing you some data from our work where we showed that stochastic conjugation in red in the context of a microtubule inhibitor, this is a monkey tox experiment, leads to significant neutropenia. However, you can get around this neutropenia by coming up with a site-specific conjugation, loading it more precisely, improving the stability as well. And that's in the blue. On the right is the pharmacokinetic graph, which is saying the same story. In a stochastic conjugation, you have payload which is coming off. The distance between a solid line and a dotted line shows you the payload coming off. It's antibody PK versus ADC PK. However, if you make a site-specific conjugation, you have these molecules quite stable, and that helps with the neutropenia. So now I want to go over, take the example of HER2 ADCs and describe what are the difference between these molecules and how, from a biological development perspective. So again, going back to HER2 is a favorite target. We all know that it's a validated target. However, just having a good target and not thinking about the technology also does not make it a successful drug. There have been 6 HER2 ADCs which have been discontinued in the clinic. And all of them have the story of their own, which we don't have time to go into. But suffice to say, many of them are discontinued because of narrow therapeutic index. So that leads to, again, going back to the principles of designing the right molecule is what makes the difference. The 2 HER2 ADCs that I'm going to talk to you briefly is the approved ones, T-DXd and TDM1, and how they differentiate from each other. So they both are on the same backbone, the trastuzumab backbone, but what they differentiate in is in the payload, the payload that has been attached, the drug-to-antibody ratio, as well as the linker. So the payload in TDM1 or Kadcyla is a microtubule inhibitor. It's a maytansinoid. The payload in trastuzumab deruxtecan is a topoisomerase 1 inhibitor. TDM1 has a drug-to-antibody ratio about 4:1, while TDXd has about 8:1. But a very important point, which sometimes gets missed, is in the linker chemistry. TDM1 has non-cleavable linker, and TDXd has cleavable linker. And why is that important? If I can hypothesize a possible mechanism here is, TDM1, because it's a non-cleavable linker, when it binds on the cell surface, it's internalized, it has to get to the lysosomes for complete proteolytic degradation of the antibody to happen to release the payload. HER2 is a very rapidly recycling receptor. It has been published that in 30 minutes, 90% of HER2 is recycled. In 5 minutes, 50% of HER2 is recycled. So if TDM1 cannot get to the lysosomes to do the proteolytic degradation to release the drug, it's possible that it is recycled back, the ADC is recycled back as an intact ADC, and it does not effectively kill the cells. Moreover, what is released in the cell has a cysteine adduct. It is not cell permeable, and it cannot cause the bystander activity. TDXT, on the other hand, is made with a cleavable linker. So the drug, the ADC binds, it internalizes into the cell. The linker comes off in endosomal and lysosomal compartment, and it effectively releases the drug before the rapid recycling of the receptor happens. Also, the payload that is released is enabled to cross the cell membrane and has bystander activity and can impact neighboring tumor cells as well. So with these changes in the linker design as well as the payload aspects, it was clear from the beginning in preclinical experiments with the studies that were done by Daiichi Sankyo that in preclinical experiments, TDXT was very effective, whether it was high HER2 expression, medium or low HER2 expression compared to TDM1. What I'm not showing you here is also what is all published out there, that in preclinical experiments, clearly TDXT shows a good bystander activity while TDM1 did not. And on the right is clinical data also published showing that TDM1 showed pathological complete responses only in tumors with high homogeneous HER2 expression. So I'm not going to show you the clinical data, which you all are very aware of, and we have the next speaker going to talk about it. But based on these, the point here is that based on the design of these molecules with the same antibody backbone, just using different linkers and different payloads may cause a dramatic difference in what ADC would effectively look in the clinic. So now with this background, we should start thinking about where can the future take us. As a field, we have come a long way, and we have 3 payload classes already out there. There is investment across the community to think about other mechanism of action of warheads which may be conjugated now. There could be a world really that we will have this toolbox of payloads and linkers and which are very effective, and ADCs may replace chemotherapy. Our target designs of targets is also improving significantly. We used to find target based on mRNA, but now as our proteomics technologies have significantly improved, and not just proteomics, but looking really on the cell surface of the cell, we are building surface proteomics databases that we can glean what exactly the receptor numbers on the cell surface of a tumor cell versus the normal cell, and start interrogating new targets in that design. And finally, if we get to the stage that we have new linkers and warheads, and we have multiple novel targets that are coming off, there will be a world, and it is actually happening, that ADCs will be combined or ADCs will be sequenced as the resistance mechanisms emerge. But to enable this vision, we need investment in few areas, and which is active area of research, especially in the clinical setting. The biomarkers, I think HER2 has clearly showed us that our biomarkers of defining where to go for is very important. We have so far relied on immunohistochemistry-based approaches, which are very good. However, we are going to go into the world of where diagnostics can be beyond human capability, and we can rely on computational pathology and AI and machine learning to help us define the biomarkers better. We also will have to understand resistance very well, and we will have a talk in the session about resistance, so I'm not going to go into a lot into detail. But simplistically, the resistance mechanism that we think will emerge for sure are around target downregulation or payload-driven mechanisms, but it's beyond that as well. We published a paper which described that when the ADC bind to the cell, inside the cell, actually cells figure out a way to change the internalization mechanism of the ADC, and they shuttle them into compartments like caveoli. So it's just so much more complicated than just thinking about payload-driven mechanisms and the receptor downregulation, and it's a lot more that we have to do in this area. And finally, the IO combinations and mechanistic combinations will be an active area of research and could be really the winning strategy here if ADCs can combine very well with IO. But there's a lot of that we'll have to learn in this aspect as well. Which payload will combine well with which checkpoint inhibitor? Today we have data emerging both from MMAE-based ADCs as well as Topo1 ADC mechanism-based ADC of combinations with checkpoint inhibitors. But again, we'll wait and see that which payload could drive the best immunogenic cell death. For example, are there differences between checkpoint inhibitors, or it will be context dependent and it will change from tumor type to tumor type. So finally, I just wanted to say that we have come a long way in the field of antibody-drug conjugates, and what we have learned in a context of cytotoxic ADCs is now being employed in the field to come up with the other modalities of antibody-based therapeutics. Immune-stimulating antibody conjugates is entering the clinic, and some of them are there, and we'll see how it goes. This field has suffered from 30, 40 years. We know we have TLR7/8, STING, biology, all plays very important role in cancer biology, but we have not been able to effectively dose them in the clinic because of significant tox issues. So could an ADC approach with this help us productively engage innate immune response remains to be seen. We are also making radioimmunoconjugates, and this field is coming back with some of the recent success, especially with the PSMA conjugate. Antibody-based approaches and the learnings that have come from ADC field and the linker technologies that are out there is helping with the PROTACs, with cell death agents, siRNA, et cetera, and this field is going to explode. So in conclusions, I just wanted to say that decades of research and technological advancements have led us to the stage that we are now have many ADCs which are demonstrating good therapeutic benefit to the patients. It's not, the rule book of ADC development is still being written, and there is not just one magic formula to do it. However, all the components of the molecules must be optimized and brought it together in the precise way to make an effective ADCs. The innovations that have happened in the design and the biomarker development and combination strategy will have to be continued to improve the success of ADCs in the clinic. And beyond cytotoxic, we are going to see this field get into other directions and learn from the ADC field on antibody-mediated delivery of other agents like immunomodulators and radionuclides. So with this, I'll conclude and take any questions.
Thank you very much, Dr. Sapra. The discussion is now open. There are microphones at the front of the room on either side. You can also use the app on the left.
I was thinking, the payload delivery to the lysosomes you're talking about. There are good examples, like in an EGF receptor. If you stimulate it, it has to go to the lysosomes. There is no recycling. We could use modalities of the signaling chemistry behind why it goes to lysosomes by default. Put that chemistry into the internalized.
Could you get a little closer to the microphone? I'm a little having a hard time.
Okay. The EGF receptor internalization, if you stimulate EGF receptor, it has to go to the lysosomes. That chemistry is encoded in the molecule. So if we decipher that and put it into these targets that we're looking at, the antibody upon internalization has to go to the lysosomes instead of staying there and coming back out, recycle back, and that's one approach. Other approach I was thinking is that your ADC resistance that you talked about and the surface availability of the target. Recently, there's a paper saying that the tumor cells metastasize in the night. There is circadian rhythm going on, meaning that we may have to examine the surface target availability in the night based on the circadian rhythm to be more effective for your ADC to work. Maybe. These are moot points to consider.
Yeah, thank you. And I think the main point of this is that we have different linker chemistries that can help us play around with the cell cycling, with the endosomal lysosomal trafficking that we can employ based on understanding of the biology of the target. So it may not be the same story for a EGFR, for example, compared to a HER2 or a TROP2, because they all understanding what is the internalization and what's the recycling mechanisms is very important.
Hi, my name is Nicholas Touroutoglu from Greece. I'm a medical oncologist. Thank you very much for this very informative presentation. I was intrigued by the way the drugs were developed. You showed on a slide the therapeutic index being one of the ways to select where you're going forward. I was wondering whether you have a clue of what is causing the ILD. Was it the linker? Is it the SN38 concentrating so much more in the lung, maybe? Would you have a clue?
Yeah, so I think I would say that this is active area of investigation. I think we are also, there is a lot of training that is going on to the investigators to how to manage it, and that's being well studied, and I would direct that to our the medical oncologist and the companies to come up with the more answers around, but it's active area of investigations and definitely a lot going on into teaching the investigators about it.
Okay, thank you.
I'm Beppe Viale from Milano. I think we are relying quite a lot on the bystander killing effect in some solid tumors, for instance, breast, and especially with HER2 low. And we know that intratumoral heterogeneity in HER2 low breast cancer is quite high. So my question is, do we have an idea of how far could the bystander killing effect go? And second, if there is any difference according to the geographical distribution of the heterogeneous cells, whether if there are two different clones that are far apart, maybe different as compared to intermingling of positive and negative cells?
Yeah, that's an excellent question. So we are now doing experiments in the lab to understand the spatial distribution of what the bystander could look like, and also defining based on our QCS computational technologies of what it could be. There is not a number in mind that we have, but it's clear that we put this different cell population in a dish, and we see bystander activity being very prominent with these kind of payloads compared to, say, TDM1, for example.
Thank you, Andrew Tutt, London. Much of what you described in a fantastic talk relied in understanding more around dynamic functional processes that are important, where the receptor density is, how it's internalized, how things are released. Do you see any potential for studying clinically circulating cells or CTC clusters to inform this going forward? Because we've got very used to liquid biopsy telling us things from DNA, but I don't see that really helping us here. Have you begun to look at that with patient materials in your studies?
Absolutely. I think we can do a lot in preclinical experiments, but we are limited with what we can do, and definitely more learning we can do in the clinic will help. We are going after pre- and post-tumor biopsy, which is always limited, and to understand that, however, we'll have an elegant talk here. We have started employing circulating tumor cells to understand both the PD, the biomarkers here, the expression of the target as well, and also it gives it gives also, the more and more data is emerging that the CTCs may also help us glean early on how the drug is working, and also may help even differentiate between a stable disease and a PR, for example, but also really understand the biomarkers.
So can I ask, you focused a lot on the specifics of the different linker technologies and how the linkers that allow for bystander effect, have seemingly made a big difference in terms of efficacy, perhaps particularly in overcoming heterogeneity. I wonder though, is it worth, can you imagine a situation where it's worth revisiting the non-bystander type of linkers, even go back to a non-cleavable linker as a way, particularly with a very potent cytotoxic, as a way to keep toxicity to a minimum and allow for your therapeutic index to be feasible?
Yeah, so I guess it's possible. I'm not saying that this is the way to go, that the cleavable linkers gives the bystander activity. In fact, Mentidium-1 is a good drug. Non-cleavable linkers are very successful in the hematological setting, for example. With a highly potent drug, a non-cleavable may work as well. However, we will then have to balance with a high potency drug the therapeutic window. What we have not been able to do is the non-specific uptake of ADCs by normal cells. So that is a big issue. And because of that, if you have a very highly potent drug and in a stable molecule, it's non-specifically taken up by all the normal cells, and then the payload is released, we may end up having the same kind of tox.
And is that non-specific?
And just to continue on that thought, one way to actually improve the stability is it's not the non-cleavable aspect, but the stability is what I was saying about the site-specific conjugation. But again, you'll have to balance that you don't now make a very stable ADC with a high potent tox, which is now giving you, it took care of, say, myelosuppression, because myelosuppression comes from payload release, but started now giving you peripheral neuropathy because you have this drug floating around for a long time.
And is the non-specific uptake felt to be FC mediated or not?
Multiple mechanisms, yes. So FC component, but also pinocytosis, hydrophobicity that the ADC gives because you have put hydrophobic payloads on it. So all of them contribute to it. It's not one mechanism.
So just altering the FC portion so that it doesn't bind to FC receptors isn't going to be the easy answer.
I don't think would be an easy answer, but on that point, I don't think we have put a molecule which FC ablated in the clinic and tested it.
Okay. So you should say I should patent that now.
And there's something which should be tried, but I don't think that will gonna fix the problem easily, else we would have done it.
All right. Thank you very much. That was a great talk.
I guess there is a question, yes, if there is time.
Dr. Wolf, I didn't see you over there.
Thank you very much. Antonio Wolf, Johns Hopkins. Two very quick questions, beautiful presentation. The first question along the lines of what Dr. Kropp was just talking about. Anything from the preclinical data that could help potentially predict the interstitial lung disease toxicity that we are seeing and related to linker technology or anything else?
This is, again, an active area of investigations. We have experiment going on in a lot of preclinical models. There is emerging hypothesis on macrophages, but there is nothing that concrete that I can share right now.
Thank you. The second question is, with the development of truly quantitative assays to measure HER2 in tumors that are not overexpressed or don't have gene amplification, and that in the past we would refer to them as triple-negative breast cancer or ER-positive breast cancer. Your preclinical data in terms of levels of HER2 in these models, any hints to suggest that measuring different levels of HER2 in these tumors will demonstrate a preferential response according to levels of HER2? Were there any data from your initial studies in these preclinical models to suggest that measuring low levels of HER2 matter at the end of the day?
So you're saying different HER2 level, what is the response of the molecule? Is there a clear correlation? I think I would say not just for TDXT, but for most of the ADC, there's not a solid answer, but if I put a receptor expression versus an efficacy graph, you will see a very strong trend. That it will be related to the density of the expression in preclinical models.
And did you have preclinical?
It will be different from payload to payload, just to be clear. But yes, for a same molecule, there will be a strong trend.
Because I'm thinking of the data with TROP2, where perhaps it doesn't seem to matter what levels you have with TROP2.
So again, I guess it goes back to it's really still complicated. What is the when you say TROP2, I imagine you're talking about sacituzumab, correct, right? So it's the mechanism of the drug is different. It's a hydrazone linker. It releases drugs over time. So you also have to look at what component of that efficacy is coming from release of the payload. It may not be a direct effect on just receptor expression, hit the target, gives the efficacy. That's why I started with the presentation. This cartoon looks very simple. This is how ADC binds, internalized, but it's a lot more than that. So if you put the component of release payload, that may help. I think what we also need is what you started saying is we need it would be great if we have a good quantitative mechanistic model developed, putting all these various components from preclinical experiments to start projecting or predicting a good ADC design early on before we put 150 molecules into the clinic. And that's something that we all are working on as a field because there is so much data out there.
Thank you.
Thanks.
Thank you very much.